Hello everyone, we are solving question number 42 of GATE 2019. It says match the instrument in group 1 with their corresponding measurement in group 2. We have 4 instruments in group 1, that is manometer, rotameter, tachometer and hemocytometer. So before matching it with their corresponding measurement, let's understand each instrument. So the first instrument is manometer. Manometer is a device to measure pressure. It is commonly a U-shaped glass tube filled with some liquid. Generally this liquid is mercury. Why mercury? Because mercury has high density. So how do we measure pressure? Pressure measurement is generally done by applied as a measurement of applied force by the liquid on a surface. So manometer utilizes that principle of hydrostatic balance where a pressure is measured by the height of the liquid it will support. So for example, the weight of a column of a mercury at 0 degrees Celsius that is 1 inch high and 1 inch in cross-sectional area is 0.4892 pounds. So that means we can say that a column of a mercury 1 inch high imposes a force of 0.4892 pounds per square inch, or you can say 0.4892 psi. So that is the measured pressure. So manometer is used to measure pressure. The next instrument is rotameter. Rotameter is a device to measure volumetric flow rate of a fluid or gas in a closed tube. So it is used to measure the rate of liquid or a gas flowing through a system of pipe at any given time. So rotameters are generally used, they are generally provide a constant flow rate measurement and can also offer optional switching capability to control the flow in a system or a process. So it belongs to a class of meter called variable area flow meters, which measure flow rate by allowing the cross-sectional area the fluid travel through to vary, causing a measurable effect. So basically here, if you could see here, the cross-sectional area varying across the tube. So once the fluid passes through this particular area, the cross-sectional area is varying. Therefore, it allowing a variable flow rate and causing a measurable effect. So through this, we get to know what flow rate is there, and we can control the flow rate in a particular system using rotameter. So rotameter are the devices that allow us to measure a flow rate of particular fluid or a gas in a system. Next instrument is tachometer, which is also called revolution counter, tach, rev counter or RPM gauge. It is an instrument used to measure rotational speed of a shaft or a disc as in a motor or other machine. The device usually display revolution per minute, that is RPM, on a calibrated analog. It look like this. It is like a gauge that is present on a particular shaft to measure the rotational speed, or nowadays we have a digital one which look like this. So tachometer is used to measure rotational speed, RPM. So the next instrument is hemocytometer, which is also called cell counting chamber. It is a tool to count cell manually. As the name implies, the hemocytometer was originally invented to quantify blood cell. The instrument looked like a glass slide, and where we have this H shaped zone, and we load a sample on this particular area and we place cover slip on this, and once we observe under the microscope, the whole this particular area look like a big square where we have four cornered square, B, C, D, E, and there are some middle square. So we don't calculate cells that are present in the middle square, we calculate cells that are present in the cornered square. And how cells look like under hemocytometer. So we use trypan blue dye. We mix the cell with trypan blue. So the viable cell that contain the intact cell membrane, they do not uptake trypan blue dye and they appear bright in color. Whereas the dead cell that has a damaged cell membrane, they uptake trypan blue dye and they appear blue in color. So when we count the cells, there are some blue color. Cells as well as some bright color cells, and that's how we calculate the cell viability. We calculate live cell as well as dead cell. So while calculating viable cell count, we count all the live cell and that divide these number of live cell with the number of large square counted. So for example, if we counted 4 large square, we divide the total number of cell with 4 into dilution factor into 10,000, and that's how we get the number of viable cell count. Likewise, we have non-viable cell count, where we first calculate the number of dead cell that appear blue in color, divide by the number of large corner square counted and multiply that with the dilution factor into 10,000, and we get non-viable cell count. And if we have to calculate the percentage cell viability, we take number of viable cell divided by total number of cell, that is number of viable cell as well as number of dead cell into 10,000. So that's how we calculate the percentage viability. So now let's match the instrument with their corresponding measurement. So manometer, for manometer it is pressure difference. So P2, so these two questions are, these A and B are out. Then we have rotameter. Rotameter is for airflow rate, that is Q4. Then we have tachometer. Tachometer is for RPM, that is agitator speed. And hemocytometer is for cell number. So answer is C, P2, Q4, R1 and S3. So answer is